Okay, so Meromit, maybe you can start sharing your screen. And so just to say, we'll start in a minute. So everybody, if you could get back and start getting ready to concentrate on the talks for the remainder of the session, that would be wonderful.
Let me see if I can. I don't know what I'm sharing. One second. This should work.
That looks much better, Meromit. So
Okay.
we're seeing the, yeah.
And I can do that.
That is perfect. Okay, so I think it's about, let me see, the clock's disappeared. Yes, it's almost 20 past. I'll just wait till it turns 20 past here and then we can get going. Oh, actually, no, it's fine. So it's a pleasure to welcome the third speaker of the session, Meromit Singer, who has a list of affiliations in the Boston area, between Harvard and Dana-Farber, and you're currently on leave at Guardant Health. So looking forward to hearing about some recent work for you, Meromit, and over to you.
Wonderful. Thank you so much. And it's wonderful to be here and great to see, you know, the community coming together, even if only by Zoom. There's still some advantages in that too. So today I wanted to talk about some work that my group has been doing recently in collaboration with Vijay Kucher's group and with Aviv's group primarily, and I'll touch on another project as well. So specifically, the main idea here, we will talk about how to use single-cell RNA sequencing coupled with TCR sequencing to get at some hypotheses about new biology that we believe to happen, and then validate them, of course. And so what my group is interested in is the balance between immune cells within a tissue and the state of that tissue. And I'm sure this audience doesn't need to be kind of pitched as to how important it is to understand the immune cell states that are within the tissue, because the change in those states can change the state of the tissue between health and disease. And specifically in my group, we focus on T cells, and within the T cells, there are the CD8 T cells and the CD4 T cells. And these T cells are, again, very important because experiments in mouse models, for example, have shown that change in expression of one main gene within T cells can change the state of the tissue as a whole, for example, by making tumors grow slower or faster. And so we're very interested in understanding this intertalk between the T cell states and the tissue state, and how the tissue state can in turn affect the T cell state. Now, of course, a main challenge in studying immune cells in tissues is the large heterogeneity between the different immune cell states and the tissue cells as well. And so to study these tissues, despite this challenge, the introduction of single-cell RNA sequencing has been transformative, because now we don't need to know what populations we have in a tissue a priori. We can take the tissue and characterize each cell within it for its RNA state, and then study the populations that we observe. And then a more recent addition to the ability to profile single cells for their RNA sequencing has been to profile the T cell receptor, the TCR, within each cell as well. And so now we have this added level of information where for these T cells that we're interested in, we see not only the RNA-seq profile, but also the T cell receptor. And I hope to convince you today that this can be very powerful. And so one thing one can do with this ability to profile the T cell receptor, as well as the RNA-seq, is to use the TCR as a barcode. And what I mean by this is the following. So for example, just to make sure everyone here is on the same page, we can have naive T cells. Each naive T cell has its different T cell receptor here, a different TCR. And once a T cell links to its antigen, once the TCR of a T cell finds the right antigen, it expands and proliferates to form a clone. And so here in this case, let's say we're looking at the blood, for example, we can have these different T cells where T cells with the same TCR we know come from the same clone. And then if we see this clone, for example, this same TCR in the gut, if we profile the gut as well, then we have indication to believe that this clone that's found in the blood is actually relevant to the immune response that's going on in the gut. And on the other hand, if we see a clone with TCRs that are found in a tumor, then we can identify this clone in the blood as relevant to the melanoma tumor in the sense, right? And so we're using the TCR as a barcode to link between, in this case, blood and specific organs, or we can do this also across organs, as I'll show. And so in my group, we wanted to use this approach to understand how systemic immunity is functioning using the T cell receptor as this barcode. And we thought we can ask two questions using this, right? One is about systemic immunity with respect to, how do the subpopulations across different organs connect? And so if we have here organ 1 and organ 2, and we have a specific cluster in organ 1 that shares a lot of its TCR sequences with organ 2, we might hypothesize that, it is this cluster in organ 1 that seeds the population in organ 2. A second question we can ask is about T cell plasticity. If within one organ, we have two separate clusters that share TCRs, and for example, in one of these clusters, the TCRs, the clones are more expanded, we can again hypothesize based on this data analysis that these cells, these T cells have this plastic capability of transitioning from being in this cluster state to this cluster state. And so I'll present to you two projects today in this context. The first one is a study that's been published this year, and so I'll just spend one slide talking about it, but I do think it's an interesting way to use TCR and single-cell RNA-seq. And the second, which I'll spend the majority of my time discussing, is a systemic immunity project in which we explored how TH17 cells function in health and disease across different organs. And so our first project in which we used this TCR as a barcode in this way was when we asked ourselves, taking tissue biopsies to study the TCR response to tumors is not an easy task. And so we were wondering, to what extent is the T cell population from tumors represented in the blood? And so what we did is we took paired tumor and blood samples from both mouse models and late-stage melanoma patients and asked, within our blood samples, how many single T cells, how many single CD8 T cells do we find that share TCRs with the tumor? And we didn't know if we would get nothing or very little or a lot. And somewhat to our surprise, we saw that a good number of T cells in the blood actually do share their TCR sequences with the tumor, and hence are tumor matching or potentially tumor directed, and that those tumor matching cells cluster in a specific location, indicating that they have a specific transcriptional state. When we compared the clone, for each clone, when we compared its transcriptional state in blood to the same clone's transcriptional state in tumor, we found that there's an increase in the state of the dysfunction of these CD8 T cells in tumor compared to blood at a clonal level. That meant that the transcriptional state of the T cell wasn't determined specifically by its TCR sequence, but that the tissue environment within the tumor had an effect on that T cell in making it more dysfunctional or exhausted. And last, our idea with this study was to enable sorting out these tumor matching cells from the blood. And so for that, we needed cell surface receptors. Because imagine how much more powered, right, your statistical analyses could be if instead of looking at the whole CD8 T cell component in blood, you could sort out and enrich for the tumor matching CD8 T cell component and study that for example, predicting response to therapies and so on. And so we did find several markers that were very good at identifying the tumor matching population. And here I'm showing you NKG2D, where we did CytSeq on this data. And so what you're seeing here is the protein levels of NKG2D. And so in this work, we proposed markers for both the mouse model we worked on and for late-stage melanoma patients in order to isolate or enrich for this tumor matching component. And this work was made possible specifically due to this ability to profile the RNA-seq and TCR in each cell so that we can identify which cell population is relevant to the tumor. Okay, and this is published work, and I encourage you all to go and read it and contact me with any questions you might have. All right, so having successfully used TCR as a barcode in this study, we were wondering if we could use it to map across organs and do a deeper analysis with, raising hypotheses about cell plasticity and migration, and then validating them with beautiful experiments that Alex has done. Oh, I'll get to that slide in a second, where I introduce my collaborators on this project. And so to quickly introduce TH17 cells to this audience, TH17 cells are T cells. They are a subset of CD4 T cells. They are primarily present in the gut during homeostasis, and they have a role in protecting against extracellular pathogens. And yes, that's all. We'll do a quick introduction to TH17 cells today so that we can talk about the computational aspects of this work. And so an interesting aspect of TH17 cells is that, as we said, in homeostasis, they're present primarily in the gut, but they are increased in the central nervous system during autoimmune disease of multiple sclerosis. And so it is very interesting to think about why this happens and how that is affecting disease. Okay, and so here, what we wanted to do is to use a mouse model for multiple sclerosis, the EAE model, and to compare the homeostasis state in which TH17 cells are present mostly in the gut to the EAE autoimmune state in which these TH17 cells migrate to the CNS. And TH17 cells have been shown by Vijay Kutru, who's hosting this symposium, as well as others, to have different transcriptional and functional states between a pathogenic state that supports kind of disease and a non-pathogenic state that's present more during homeostasis. And so the main questions we asked to guide us in this study was, what is the baseline heterogeneity of TH17 cells across organs? So here, how does baseline heterogeneity change upon EAE disease? And upon EAE disease, where do the TH17 cells in the central nervous system come from? Because they're not there before disease happens. And this project was initiated and thought of by an incredible graduate student, Alex Schnell, who's a graduate student in Vijay Kutru's group. And so when I was a postdoc in Aviv Regev's group, Alex approached me and said, I have the coolest project ever. You have to work with me on this. And it wasn't hard to convince me because her idea was indeed, very big and innovative, as you'll see. And so I started working on this project with Alex during my postdoc. And when I started my own group, Linglin, who is another incredible graduate student, joined to work on this project as well. And so this is really, I feel, one of these cases where it's been just a super fun collaboration of individuals with different backgrounds coming together to study the system. And so what we did is we took mice without disease first at homeostasis and the different organs from these mice. and sorted out TH17 cells by using reporter mice for the IL-17A gene. And so we had both fate reporters, meaning a reporter determining whether this cell ever expressed IL-17A, and an active reporter telling us whether this cell is currently expressing IL-17A and hence is a current TH17 cell. And we took these single cell sets from all of these different organs, and we first wanted to ask what are the main determinants of variation between these populations. So how do these populations differ from each other if we just throw them together, data coming from, of course, multiple different mice, and do this exercise of dimensionality reduction and asking what are the main, how do these cells cluster? And what we saw, potentially to our surprise, we weren't necessarily expecting this result, is that when you take all of the TH17 cells and put them on a UMAP or clustering, you see that they cluster by their organ of origin, right? So this told us that the main determinants of variation in this data, the main determinants of the differences, the transcriptional differences between the TH17 cells from different organs originate based on which organs they came from. The proliferating set and a TH17 Treg-like set were the exceptions, but aside from that, you can really kind of infer which organ a cell came from based on its transcriptional state. And we looked at this further by looking at a gene by gene level and identifying genes that were upregulated in specific tissues compared to the spleen. And interestingly, we found these different gene modules where we had a module that was present across all of the different tissues of upregulated genes compared to the spleen. But then we also found tissue-specific upregulated gene modules, indicating kind of what we see here, right? That different TH17 cells are distinguished from each other based on what organ they came from. And so interestingly here, you see that when you just take the cells together, then as I said, there are main differences based on organ of origin. However, interestingly, when we did a different exercise and we clustered each of the organs separately into, transcriptional clusters, and we functionally annotated these transcriptional clusters based on known biology, we actually found similarities across organs with respect to the transcriptional functional states that are found within them, right? So when you look globally and compare organs to each other, you see these transcriptional programs that distinguish a TH17 cell depending on which organ it came from. But when you look within organs, then you find similar transcriptional states that are present in subpopulations of the TH17 cells across organs. And what you can see here are these different organs and the different clusterings that were done in those organs. And when you look at the upregulated genes of the different clusters and cluster the clusters based on that, right? These here are the different clusters from the different tissues. You find, for example, that in each of your tissues, you get a proliferative cluster. And in some of your tissues here, right, you get a T follicular helper cell-like state. And so I just think it's nice that you can kind of look at single cell expression data with these different angles and identify different similarities or differences across your data sets. Okay, I'm going to skip this because I'm a bit short on time. And then we wanted to understand how the TH17 cells and states that we've characterized in homeostasis change during EAE disease. And so we did the same exercise in which Alex dissociated, the different organs from mice and sorted out the TH17 cells. But this time she did this at initiation of disease. And of course, because this is a computational conference, this was not done after the fact, right? In each of our experimental batches, we had both homeostasis mice and mice with disease. It's just easier to present things this way. And so when we took the TH17 cells from the diseased state, we again saw this distribution based on organs, right? So the fact that a disease state occurred did not change this characteristic of the TH17 cells where they're different based on the organs that they're in. And we were interested here now to use this ability of looking at the T cell receptor as a barcode. And we asked ourselves, where do the TH17 cells in the central nervous system here, the CNS, come from? Because remember I told you in homeostasis, we didn't have them. And so we decided to look first at our most expanded clones across these different organs. So for each organ, we took the 20 most expanded clones and we asked how are they distributed across the organs. We actually thought that we'd find a considerable number of clones from the gut that transition into the central nervous system. However, what we saw was something different. And so here you can see just for the most expanded clones across our tissues here, each row here is a single T cell clone. And you can see its relative abundance in each of the different tissues. And it's not easy to see here. Here are the different tissues below. It's not hard to see, that you have these two kind of separate clusters of distribution of the different clones, right? You have a cluster of the spleen, the central nervous system, and the draining lymph node, and you have a separate cluster of the gut organs with some overlap with the spleen. We were somewhat surprised by this, as I said, but this looked like a strong result. And so we actually looked at this globally, not only based on the most abundant, most expanded clones. And for each pair of organs, we asked, what is the extent to which you're linked in your TCR expression, in the extent, in the clones that are present in each organ and the extent to which they're expanded. And again, when we look globally across all TCRs, we see this cluster here of the CNS, the spleen, and the draining lymph node, and we see a separate cluster of the gut organs, the colon, the small intestine, parapatches, and mesenteric lymph nodes. And we also see that the spleen, while it's mainly clustered with the CNS and draining lymph node upon disease, it also is well connected to the gut organs. And so we asked ourselves, okay, it looks like the spleen is an important link here between what's going on in disease and the gut organs, right? And this is also in disease, but between the populations of TH17 cells in the gut and the clones that are found in the central nervous system. And so we decided to zoom into the spleen and take a closer look. And so what I'm showing you here are TH17 cells from the spleen during EAE. We wanted to study the TH17 cell population in the spleen during EAE. And when we clustered this population, these two main clusters came out. And we were wondering what the differences are between them. So we generated upregulated genes for each of these clusters and asked how they connected to previously known biology about the TH17 cells. And what we saw was that these two clusters were very different in their TH17 known kind of pathogenicity expression landscape, where when comparing to these known and established gene signatures, it became very clear that this red cluster down here is associated with pathogenic TH17 cells, so with TH17 cells that are found and known to drive disease. And this blue cluster up here was associated with transcriptional states of non-pathogenic TH17 cells, TH17 cells that do not cause disease. And when we looked at the expansion rates using the TCRs here, we saw that the pathogenic cluster was more expanded than the non-pathogenic cluster. And so we started thinking, it looks like a lot is going on in the spleen during this disease state. And specifically, this pathogenic cluster is active and has these expanded clones in it. And so we asked, can we infer migration and plasticity, like I've shown you in the beginning, using the TCR data from these two clusters? And to do that, we looked at the TCRs in these different clusters and asked for each cell, is the TCR found in the CNS and the central nervous system or not? And what you can see here in green are cells whose TCR was not found in the CNS. And in pink, you can see TCRs that were found in the CNS. And you don't really need a complicated statistical analysis to see very clearly that the cells from this pathogenic TH17 cluster in the spleen are found in the CNS. Their clones are found in the CNS. And so it really looked to us like this population here in the spleen and not the non-pathogenic population was seeding the TH17 population that is found in the CNS. And so this is what I just said. And so we were wondering, okay, this shows the connection to the central nervous system. How are these two clusters connected to the other organs, to all of the other organs? And so we made this map here where you can see this is for this spleen 0, the cluster up here, the non-pathogenic cluster, and this is for the pathogenic cluster. And what you see here is a score of how associated this cluster is to each of the different organs based on the clones and their abundance. And you can see something really interesting, that the pathogenic TH17 cluster is overlapping, let's say, I don't want to interpret this, but potentially seeding the TH17 populations in the CNS and the draining lymph node, but not in the gut organs. Whereas this non-pathogenic spleen 0 cluster is potentially contributing to the TH17 cell populations across all of the organs somewhat equally. And this brought us to hypothesize, right, that this is indeed, that there are these two systems in two different populations in the spleen that have very different overlap across organs. And Alex went ahead and validated this using transfer experiments to generate beautiful results, but I'm not going to show them here because I wanted to show a different validation. So I'll be done in 1 minute. And so then we asked, can we infer about cell plasticity using TCRs, right? Specifically, is the non-pathogenic population turning into the pathogenic population in the spleen? Because as we've seen, this pathogenic population is more expanded, and we saw some overlap between the T cell receptors present here and in the pathogenic population, but in the pathogenic population, they were more expanded. And so we hypothesized about this plasticity. And indeed, Alex validated this plasticity as well using transfers. And so what you can see here is that when you transfer, we sorted out for these two populations and identified markers for them. And so here you can see that when you transfer the blue population, when you transfer the non-pathogenic splenic population into EAE mice, you can then take out, right? You can then find within these mice both the pathogenic and the non-pathogenic population. However, when you transfer into an EAE mouse the pathogenic population, the red population, you then only find that population present within your EAE mice, within the spleens of these EAE mice. And so this is really showing us this, hypothesis that was generated from the ability to use TCRs as this barcode to generate an understanding about TH17 plasticity in the spleen during disease, which we thought was pretty cool. So we can draw this arrow here without the question marks. And that's it. So I told you about these two projects. I want to emphasize again how wonderful of a collaboration this was, and to thank specifically Alex and Linglin for their tremendous amount of work throughout this project. And of course, Vijay and Aviv for being incredible collaborators as they are. I think I'm done. Yeah.
Thank you, Meromit. Excellent. Really, really enjoyed that. And we have several questions in the chat. So let me start by reading out one from Scott Tyler. So how do you account for the differences between the number of T cells observed across batches / patient / organ samples, since basically all measures of diversity, clonogenicity, etc., are directly determined by the number of T cells observed? he's always favored random downsampling of each sample to the lowest number of T cells because it's statistically safe and robust to false positives.
Yeah, so let me break that up a little bit. So let's first talk about batches, regardless of T cell receptor analysis, right? So first of all, right, we have different batches in this experiment, of course, and we want to make sure that our observations are not driven by batches. And we actually spent a long time, like, many months thinking and making, thinking about how to construct the data such that our results are not driven by batches. And at the end of the day, we did downsampling. We added batch as a covariant for any, upregulation, downregulation of genes that we did. And we regressed out batch to generate gene expression levels, which we then use dimensionality reduction on and so on. While we were doing all of that, batch correction methods were developed by yourself, John, and others, and Rahul. And so we ended up using the kind of out-of-the-box methods to push this data for the visualizations and making sure that they aligned with the analyses that we've done before that. And for the differential expression analyses, we did keep batch. We did correct for by adding batch as a covariate, as a fixed effect. So that's about the batches. We spent a lot of time making sure that our analyses are robust to that. With respect to TCR analyses being affected potentially by differences in cell counts across batches, again, yes, that's a very big problem that makes you jittery potentially about your results. We again approach that by doing downsampling to do sanity checks for ourselves that what we see is true across batches. And we also accounted for this in the TH17 work by experimentally validating the hypotheses that came up from the data, right? So essentially consistency across batches, downsampling where possible. Sometimes it just hurts your statistical power too much. And there's nothing like experimental validation on new batches to show you that your hypotheses are correct. So.
Excellent. I mean, I guess before we go on to the next question, just slightly related, were the results completely consistent across mice here?
Yes, so the results were consistent across mice. That's an interesting a fun, feature of working with mice, which I definitely cannot say about the patient data we worked with. Results were consistent across mice. Sometimes for some mice, we weren't powered to identify the, profiling that we saw in other mice, but they were definitely leaning in that direction. We never saw things that were statistically significant in different directions across different mice. Yeah.
Excellent. Okay. So then the next question from Tianli Zhao. So they ask, is the reason that CNS clones mostly mirror splenic clones due to the immunization model with CFA, which might be expected to drive pathology starting in the peripheral lymphoid organs? Would you see stronger mirroring between CNS and the gut in MS patients?
Okay, I missed the first part of the question.
It's in the Q&A bit. If you open that up, Meromit, you can see it.
I'm Zoom challenged, but okay, here we go. Okay, here we go. Which?
Tianli's question.
Okay. Is the reason the CNS? Okay. Oh. Okay, that's a great question, especially because I don't completely know how to answer it. And if Vijay would want to have a go, I would love to do that. We did look for some parallels with human TH17 transcriptional states and did see parallels, but with respect to specifically this, whether this type of clonal distribution is specific to the mouse model that we've looked at due to the biology of this model, I can't answer, and I'd love for Vijay's input. Yeah.
Okay, so I don't spy Vijay at the moment, so I will.
It's indeed because of immunization. What happens is you're generating a large series of clones. and there's another trajectory of T cells which you are talking about, which are of non-pathogenic origin, and they seem to go to gut. So it is CFA immunization is helping you expand the clones as an adjuvant.
Okay, excellent. So actually we can have a, oh, can I make someone speak? No, I can't do that. How do I make someone speak in this version of Zoom? I don't think I can. So Samuel, you'll have to, I think, type your question, because I don't think I'm allowed to enable your mic, or I don't have permissions to enable you to speak. Perhaps one of the co-hosts can do that. No. Oh, okay. We'll write in the question. So whilst we're waiting for that, Avi Srivastava has a question. So I concur with the first part. Amazing talk, Meromit. In the first plot of clusters across normal samples, and was wondering, did you zoom in on the populations that are not common across organs? And curious if that could reveal organ-specific changes relative to disease states.
Yes, so that's an excellent point, and we spent a lot of time looking into that. There are differences in some of these populations for sure, but they didn't seem to change much upon disease, right? And so we did see, we did, you know, as you always do with these big data studies, we have a list of hypotheses regarding change in TH17 cell populations within organs, and asking whether they are functionally relevant, right? Or kind of some feature of this. We started following up on a few, but the main kind of differences were seen in the spleen. And so we started by looking into that.
Excellent. So I think I now have permission to let people talk. So let's have a live question from Connor Lambden. Connor? Are you there? The microphone is muted, I think. You have your hand up. Or not, in which case we'll go to an anonymous attendee question. So, which is simply, how do you define similarity in TCR clonal populations across tissues?
Yes, thank you very much for this question, especially for this crowd. I'd love to share that. And so Linglin tried a few things, and the approach we felt most comfortable with is simple at the end, but I think elegant. And so for each, you have two populations of interest, right? And so for each population, for a given population, you have your different clones, and you have a cell count for each clone, right? How many cells from that clone did you see in this population? And so you just take the relative frequency of each clone. So essentially you have your counts, you divide them by the sum, your n. And so you have this kind of multinomial variable, right? Where essentially the probability of sampling a cell from a specific clone from each of the specific clones, right? So you just take the counts, divide them by the sum of the cells in that population, and you have this for your two different populations. And so for each clone, you have the relative frequency of that clone in population 1 and the relative frequency of that clone in population 2. And then we take what we call the Bhattacharya distance, and I hope I'm not butchering that, which is the sum of the square root of all of these relative frequencies multiplied. And so the Bhattacharya distance, if you know it, great. If you don't, Google it. It's a simple measure that's between 0 and 1, and is essentially the distance between these two relative frequency distributions. And so when you saw those different circles of the distances, it was just a visualization of that value between 0 and 1 that we got.
Excellent. So I think with that, we can probably say thank you again, Meromit, for a lovely talk, and thank you for everybody for the questions.